Okay, so we are coming to the conclusion of this very interesting conference. So for me, I learned a lot today. I hope you also find a lot of useful hints for your daily work and for the future. So how do we do now? So the idea is that we have some closing key remarks by the session chairs. So I will start it and ask my co-chair, so Blanca Landa, what do you think, Blanca, from our session? What are the key results of our session and what are the next steps for Xylella?
Yeah, I think that it was clear that the European legislation and the regulation has been moving fast as we get more knowledge on the different Xylella fastidiosa outbreaks. At the beginning, we have a fixed idea that every outbreak was the same. And thanks to the different projects and research ongoing in Europe, also the work of EFSA, the different legislation has been moving and implementing in a different way. What is clear is that this work has to continue. We have to provide more answers that need to, more answers to the different questions that still remain, and ensure that the legislation will move in the same direction. We saw how in Italy the situation is changing. Maybe there is some hope, and we can see in the future how this reduction of symptoms can be even better. We need to learn how to implement the different measures in an integrated way to be able to try to stop this progress of the epidemic in Italy. Also, we saw the importance, not only in this session, but in others, of the use of genome analysis to understand what we have in the different outbreaks, how the movement of Xylella could happen in the past, and this can serve as a tool to avoid this happening in the future. We also saw that we need to understand not only the genetic of the plant, but also the microbiome of the plant and the roles that can play in developing Xylella symptoms or avoiding them. Finally, what is important, we are always focusing on the countries that are affected by Xylella, that are more on the south, but we cannot forget that the northern regions in Europe are also under at the threat of this pathogen. So maybe the subspecies and the environmental conditions are so different that we don't know yet how they can evolve in case there is an introduction. So research needs to study those pathosystems, and we should not forget them. Yeah. So I will pass now the floor to the second session moderators.
Thanks, Blanca. For our session on the Xylella fastidiosa resistance and control, what we have seen, there is several research with some putative control in the pipeline, in the lab, but also in the field with some tolerant cultivar. Remember the talk from Ophir with some almond variety, more tolerant to Xylella fastidiosa. Also the talk from Pasquale Saldarelli on olive, if I'm not wrong, it was Lecchino cultivar. And also with some talk on grapevine resistance induced by LPS, or also with transformed rootstock. So there is something in the pipeline. What we need to think also is to, when we have some compounds like LPS or peptide to apply it in the field, is to make upscaling of this kind of compounds to really spray them on a lot of plants as foliar spray or to inject them. And I would like to give to Maria if you have some other conclusion to say.
Yeah, just on the part on the genetic resistance in olive and in almond, you heard the work that is going on in Italy and in Israel. For olive, I have to say that we suffered in the past year the gap of knowledge on the genome olive to advance the research on genetic resistance. But even we still have only two cultivars so far available as resistant genotypes. We know now that because of the screening, the large screening test that has been done in the past years, and the work done in US with only 3 cultivars is also another example of how difficult it is to find natural resistance in the cultivars that we know. It is important now to have the knowledge that has been gained about the progenies derived from the resistant cultivars, and especially to know that when the resistant cultivar is crossed with the highly susceptible cultivars, we have a progeny that display a different behavior, and this is important to start the genetic mapping, the quantification, the QTL markers identification. So this is the work that has been started now in Bexil, and at Italian level will be intensified with 2 important projects that the Italian ministry have supported. So even if we still don't have more cultivars, we know that we must invest in the breeding programs, and the breeding programs can benefit for this knowledge that we gained, and it must also benefit from the activity of the proximal sensing, because what we need to screen these crossbred progenies is fast markers, biological markers to screen susceptible and resistant. And in this, for sure, all the work that is going on phenotyping and proximal sensing will be very important.
Can I also ask whether do we have all the resources available for working on the germplasm for resistance, for olive, for almond? I mean, do we have the collections of all the variety of Mediterranean, the olive genome? Is this available that we can use?
Well, we have some few data, but I would like also to know why some cultivars are more tolerant than the other. Is it the genotype of the plants, the physiology, the plant microbiome, a lot of things together? And when we are talking also about cultivar more tolerant, until when? With the evolution of the pathogenic bacterium. So we have some dispersed data, but it will be very interesting to know more about this. But it needs a lot of years of research, and there are some people working on genetics here. They can help us to know more, but maybe, you know, it's not a single gene, it's a lot of gene cluster, a lot of factors that can influence the tolerance of this different variety. Yeah, and maybe we can find also more tolerant variety somewhere, I don't know, yeah.
And just if I can add that the information that Pasquale brought about the identification of putative susceptible genes is also very important and very important aspect that can be exploited with the new genome editing techniques to most likely try to confer resistance to the susceptible cultivars.
So now we ask Rodrigo and Astrid. So on vectors and vectors control and biology, what do you think is the key result from today, and what are the next steps?
So I think maybe to follow up on what Rodrigo was saying just at the end of the discussion, I was struck by the fact that we are able to follow the development of Philaenus plumarius almost day by day with this progress we have done with modeling. But despite of this, we are not able to clearly give some recommendation to how to manage those different populations of the most important vector in Europe. I'm not quite sure if we could have a European scale strategy. I'm not sure. Maybe some regional scale strategy, because as it was stressed, we have some differences between what we observed in Italy, in Spain, and also in France and Corsica. The behavior is a little bit different. I'm not quite sure even that maybe the good stage is the infill stage. I'm kind of thinking that we could concentrate on different stages to set up some management experiment. And then I think what struck me as well is that, so we have more and more data on Philaenus primarius and a little bit also on Neophilaenus campestris, but the other vectors are almost ignore. So we had a presentation at the beginning of the conference by Lena on Afrophora. So we have a little data on Afrophora, but not that much. So we still focus really on, which is understandable because this is the most important vectors, but we have not a lot of clue on the community of vectors that are occurring in Europe. So yes, this is maybe what I retain from what we have seen today.
I would also add a couple of things. First, I do agree that we know a lot about Philaenus. If we can implement some sort of big trial and see if, put all these data together and come up with some management, or at least figure out when you can actually manage the nymphal stage or not, remains to be seen, but we may be there already. What I wanted to say is we had 2 talks, not from Europe, on Xylella and olives and transmission, and that's kind of cool to see, and I think that there is a lot of potential there for understanding, in California, the differences between multiplex and fastidiosa, for example, where multiplex does not cause disease, but is kind of present in olives in California, but the fastidiosa, no, the pauca, sorry, from Italy causes disease, so some interesting things that could come out of that. The same thing is true from Brazil. What I will note, which is, and correct me if I'm wrong here, but the one thing that seems to be really missing, and not only from the conference, but in general from Europe, is transmission research. There is very little effort in connecting what's going on in terms of the vector and transmission and spread in the field, in terms of experimental manipulations and trying to understand the transmission process. We had talks, the talk from Brazil and the US included transmission data, or the US at least half the transmission process. The other ones really focused on the insects. And there is transmission work going on in Europe, but it seems like there should be a whole lot more than at least is getting published. One particular component of that, of course, is that there is very little or no transmission work in Europe on the Xylella vector interactions, which is challenging, of course, but a lot has been done, and there's very little or nothing that's being published. So this, to me, of course, as someone who studies that, it's a big gap, because without transmission, you're not going to have spread of any kind.
Yeah, just before going to the last session, I would like to tell Rodrigo, don't forget that you have Xylella everywhere in California, mainly. This is a pathogen of quarantine pathogen in Europe. So to work with transmission and infective vectors, you need very strong quarantine facilities. And I'm sure even I have the very best in the world, I would never work with infected vectors in a place that you can have an escape and get a problem. So it's not easy to work with transmission. That's the key point, yeah.
I'm not disagreeing. I'm just saying that the knowledge gap remains regardless of the limitations.
So the last session, I pass the floor to Valérie and Claude. Thank you.
Yeah, what was interesting is that now we have tools to go at the really very fine level for epidemiological surveillance, like with the plasmids. Really, you can track the populations. And also, we have some tools to know if the plants are infected, new tools. And also, without symptoms, we start to have tools for surveillance and to help the risk managers to know maybe in the future what plant to choose when they are doing their sampling. I was was also interested by the multispectral and the sensors, because it's tools that maybe could not only be used for surveillance, but for resistance testing or for biome control evaluation, which could be a little broader. And it's also interesting, all these tools to really help the risk managers to know how to organize their surveys, what to sample, and working together with people from different domains who can help and improve these surveys. Do you have?
No, maybe what I would like to say, I don't know, some of you attended the conference in Corsica or the one online, and what was striking to me is that it seems there is a move. We have no more presentations on PCR and so on, but the presentations are much more on coordination of on robustness and harmonized pest surveillance, and that is key, I think, and it's nice to see that we are going along this road. Progress also on some technologies which are almost ready, as mentioned, for proximal sensor or high-resolution multispectral satellite data, but still for early detection, we could move a way forward on this, and it would be very nice to bring these nice technologies also to the sampling on the ground. And we have seen very nice surveillance for the delimiting survey, for example, in France, or the approach in Alicante in Spain. But these are also hotspots in contaminated country. And my fear is that, are we doing enough? Are we not looking only at the top of the iceberg where we know that the bacteria is? And for all the places where the bacteria could occur, are we doing enough? And we had no presentations offered from countries where Xylella is not so much reported in the Mediterranean country. And I have questions about this. Of course, maybe it's just that they are surveying and there is nothing to say. But it would be nice maybe to have a discussion and look at what is done also there.
In fact, European Union has set up a very coordinated surveillance with all the member states, but this is now extended also to the neighboring countries. So in the network was mentioned before by Alice, we are a member from the Balkan country, the pre-accession country, the country which are neighboring to the EU. Now they are, in September, the same tools will be trained in Africa. In Nairobi, there is an international meeting, and then an international African meeting where the tools for RIPAS will be present and the inspector will be trained. So we're also trying to do outside of the core groups or the affected countries. So that, and I think that part is quite advanced. What we all notice, I think that now we have tools are being really finalized. So now we have to think about how these tools can make the life of our plant health manager, farmers easier. So I think we can now go to close this section, and we have a few slides that we will show together with Blanca, and I don't need a double microphone. I think that's too much. So time is really running, and now the ICPP, the International Conference of Pathology, is also starting at the same time. So just to recap what happened in the 2 days and yesterday, we had 200 participants registered from 41 countries. I think if you look in the room now, we are probably around 180, 160 in this moment. 23 presentations, 51 posters. This is for the first time this meeting was hosted by the International Conference of Plant Pathology, so we tried to have this meeting paired with a scientific meeting and was streamed. So I don't know if we have any statistics, but many people is watching online, and then all the video will be available in the coming days also as a record on the EFSA website. So for me, it was a very nice meeting, and I think we need to thank all the participants, the speaker, the poster presenter, the participant in Lyon online, and then all the scientific committee and session chairs, organizing committee. And here I really would like to ask you to applause for Alice Del Bianco, Alice Cantino, that have really spent so much effort for this. And then I don't know how good you are with social media, but there were a lot of Twitter from this conference, and there's a team that's working on this: Alexia, Evelina, Marina, Giulia, Sara, that's remotely from Parma, and I also would like to thank them very much. Particularly Giulia was the movie director for our Twitter. And then we also had people working behind the scenes, so Pomilio Bloom, which is the company working for EFSA to organize events, and then here we are ICPP and all the ICPP, the Europa teams that really works a lot for us for this meeting. So thanks to all. And now I think we should have an announcement, because we had already 4 Xylella conference meeting, and we dare to tell you that we organized the 5th one, and this will be in collaboration again with Bexil and in Italy. Yeah. So in 2025, probably in the second part of the year, we still don't have a date or a place, but we'll work for it. So thanks a lot for all, and you will receive a survey for satisfaction about the conference, so please also fill it, and have a good return home for those who leave, and a good stay in Lyon for those who continue with the ICPP conference.